Hello, welcome back. In this short video, we're going to be covering the flow of genetic information. So go ahead and get yourselves a paper and a pencil, and you can follow along with this diagram, which is rather simple, but it's meant to illustrate a very important concept, one which we will be revisiting again and again throughout the course of this quarter. All right, so the first thing I'm going to do is I'm going to draw a large cell. All right. So here's my large cell. It kind of looks like it has an egg shape to it, but this line which delineates the outside versus the inside is meant to illustrate the plasma membrane of the cell. So that barrier which separates the extracellular environment from the intracellular environment. Okay? So as we've learned, inside of eukaryotic cells exists a nucleus. Okay, and the function of the nucleus is to compartmentalize the genetic information. All right, so inside of the nucleus, we find all of the DNA that is bound up in chromosomes. Now in humans possess 46 chromosomes. So I'm just going to write the word DNA right here. Okay, and I'm going to draw, as a cartoon for DNA, a little double helix like that. All right, now DNA remains inside of the nucleus and it cannot leave the nucleus. All right, so we can think of DNA kind of like the genetic blueprints for everything that makes up a cell, right? So I'm pretty sure that you guys have some basic idea that DNA is the blueprints of life. Well, what does DNA actually encode for? What kind of information does it store? Okay, so inside of this nucleus, we have DNA where it remains, but DNA has to be copied before those instructions can be carried out by the cell to make the stuff that the cell is made of. All right, so DNA, we say, is transcribed into a copy of the DNA. Now, here, DNA is illustrated as a double-stranded double helix, but the copy that is made of DNA is single-stranded. Okay, see, I've written here this blue squiggly line, a single strand, and this material is another nucleic acid, RNA. Okay, so ribonucleic acid. Okay, so if DNA is the blueprint, RNA is the instruction. Okay, now what happens with the RNA? Well, we said that the DNA cannot leave the nucleus, but RNA can snake out of little pores in the nucleus that are called nuclear pores. Now, these are little channels or little gateways that lead out of the nucleus, and RNA can snake out into the cytoplasm, or the liquidy matrix inside of the cell. Now, it's in the cytoplasm that the RNA is translated, all right, into a new type of a macromolecule called a protein. So I'm going to draw some structures which we're familiar with. All right, so I'm going to draw some alpha helices, some beta pleated sheets, more beta pleated sheets, more alpha helices, and you can see that this kind of looks like a mess, but this is supposed to represent the tertiary structure of a protein. So an amino acid sequence of, all combined into a polypeptide that folds into the secondary structures, the alpha helices, the beta pleated sheets, that folds into the functional tertiary structure. Okay? So it's the protein that is the product of genes. All right? So that's a really important concept that we're going to revisit. And the two processes that are carried out in order to go from DNA to RNA to protein are entitled transcription and translation. Okay, and we're going to go over those two processes in much greater detail as we go further in the quarter. But first, an analogy. Okay, now an analogy is a story where I try and make sense of something and relate it to something that you guys might be more familiar with. All right, so